Hi, good evening, everybody. Welcome back to Daily Lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I am Dr. Bobby. We're on to section 3 of molecular biotechnology and on to the chapter which deals with engineering plants to overcome biotic and abiotic stress. And going forward, we have on the chapter of engineering plants to overcome biotic and abiotic stress. The chapters of interest, or the topics of interest, are insect resistance, increasing expression of the beta-thuringiensis protoxin, other strategies for protecting plants against insects, preventing the development of beta-thuringiensis resistant insects, virus resistance, viral coat protein mediated protection, protection by expression of other genes, herbicide resistance, fungus and bacterium resistance, oxidative stress, salt and drought stress, fruit ripening and flower wilting, summary and review questions. Right, the principal objective of plant biotechnology is to create new varieties of cultivated plants, cultivars. The majority of the initial studies of transgenic plants have focused on developing strains that give better yields. The majority of the initial studies of transgenic plants have focused on developing strains that give better yields. Genes that confer resistance to insects, viruses, herbicides, environmental stress, and senescence have been incorporated into various plants. A considerable amount of this work has been commercialized and has been the subject of much public scrutiny and discussions. Some of this work is discussed below. Insect resistance. The genetics engineering of crop plants to produce functional insecticides makes it possible to develop crops that are intrinsically resistant to insect predators and do not need to be sprayed often, 6 to 8 times during a growing season, with costly and potentially hazardous chemical pesticides. It has been estimated that in 2007, that the amount spent on chemical insecticides worldwide was approximately $15 billion to $20 billion. The cost of maintaining such genetically engineered insect-resistant crops is lower than that for non-resistant crops. Moreover, biological insecticides are usually highly specific for a limited number of insect species, and they are generally considered to be non-hazardous to humans and other higher animals. In addition, by reducing the damage to plants from insect predations, a corresponding decrease in the damage to plants from a number of fungal diseases should result, since many pathogenic fungi often invade a plant either together with or as a consequence of insect infections. Several different strategies have been used to confer resistance to insect predators. One approach involves a gene for an insecticidal protoxin produced by one of several subspecies of the bacterium Bacillus thuringiensis, as seen on chapter 16. Other common strategies use genes for plant proteins such as alpha amylase inhibitors, protease inhibitors, and lectins that have been shown to be effective against a wide variety of insects. After an insect ingests one of these inhibitors, it is not possible to digest food, that is plants, because the inhibitor's interference with the hydrolysis of starch or plant proteins. Thus, the insect will feed less and eventually die, increasing expression of the beta-thuringiensis protoxin. Beta-thuringiensis protoxin does not persist in the environment, nor is it hazardous to mammals. Thus, it is a safe means of protecting plants. It is both simpler and less costly to express the genes for beta-thuringiensis toxins in plants than to spray beta-thuringiensis preparations onto the surface of the plant. This mode of insecticidal toxin delivery limits the environmental distribution of the toxin and avoids problems associated with the spraying beta-3-enhances preparations, such as limited environmental stability and the timing of the toxin application. The scientific challenge in utilizing the beta-3-enhances toxin is to create a transgenic plant that expresses the synthesizes a functional form of this prokaryote insecticide at sufficient levels to prevent damage by insect predation. In initial experiments, the beta-3-enhances subspecies Kritstaki insecticidal protein genes CRISPR-1AA, 1AB, AB, AC were not particularly well expressed in plants. This is problematic because high levels of expression of these insect control proteins are needed in order to produce commercially viable insect-resistant plants. To raise the level of the expressed protein, scientists truncated the gene so that only the N-terminal portion of the insecticide protoxin, the part of the protoxin that contains the toxin, see chapter 16, was produced, and inserted a strong plant promoter to direct gene expression. Under these conditions, there was a significant increase in the level of insecticidal toxin produced, affording transgenic plants some protection against damage from insect predation. The minimum sequence that encoded toxin activity had to be determined. To this end, the amino acid sequence of protoxin from various strains of beta-3-enhances were compared to determine whether there is a common insecticidal toxin domain. This analysis showed that the N-terminal portion of the protoxin molecule is highly conserved, that is 98%, and the C-terminal region is more variable, 45% conserved. Further work showed that all of the insecticidal toxin activity resides within the first 646 amino acids from the N-terminus of the 1156 amino acid protoxin, or the segment of the protoxin gene that encodes the highly conserved amino acid sequence was cloned and expressed in bacteria. The shortened protein was as active as the native protoxin form in protecting plants against lepidopteran insects in laboratory trials. In both greenhouse and field trials, transgenic tomato plants that express the short form of the protoxin were protected to some degree against damage caused by tobacco hornworms, Manduca sexta, tomato trust fruit worms, Heliophysia, and tomato pinworms, Kefera lycopersicella. The extent of the protection was not the same for each of these insects, nor was it complete. The transgenic plants were protected to some extent from damage caused by tobacco hornworms and tomato fruit worms, and to a lesser degree from the damage by tomato pinworms. A combination of a low dose of chemical insecticide and production of the protoxin by the plants increased the level of protection afforded by the protoxin. In an effort to dramatically increase the level of expression, an isolated insecticidal toxin gene was modified by site-directed mutagenesis to change any DNA sequences that could inhibit efficient transcription or translation in a plant host. This partially modified gene had a nucleotide sequence that was 96.5% unchanged from that of the wild-type gene and encoded the identical insecticidal toxin protein. Transgenic plants that expressed this partially modified sequence produce a tenfold higher level of insecticidal toxin protein than the plants that were transformed with the wild-type gene. Subsequently, a fully modified version of the insecticidal toxin gene was designed and chemically synthesized. This fully modified gene contained codons more commonly used by plants as opposed to those favored by gram-positive bacteria such as beta-thuringiensis. This gene was also modified to eliminate any potential messenger RNA, mRNA secondary structure or chance plant polyadenylation sequences that might decrease gene expression. After modification, it had a G plus C content of 49%. The wild-type gene is 37% G plus C. A nucleotide sequence that was only 78.9% identical to that of the wild-type gene. And now we're on to a milestone note: light-inducible chloroplast-associated expression of a chimeric gene introduced into Nicotiana tabacum using a Thai plasmid vector. Courtesy to El Herrera, Estrella, Van der Broek, Mainhot, Van Montego, Shell, Timko, and Cashmore. Courtesy and thanks to them for this milestone note. After researchers had established the Thai plasmid system as an effective means of transforming many different plants, the attention turned to the development of procedures for the expression of foreign genes in plants. Initially, most of the genes that were introduced into plants cells were under the transcriptional control of either the literally strong constitutive 35S promoter from cauliflower mosaic virus or the nearly as strong constitutive promoter for the nuclein synthase gene that is encoded within some tDNAs. However, the development of plants with useful new and modified traits often requires that a specific protein be expressed only in certain tissues. Example, leaves or roots, or only at certain times in the life of the plant. Example, during early seedling development, during formation of or high temperature stress. As a first step toward the development of plants that express foreign genes in a tissue-specific or time-specific manner, Herrera, Estrella et al. Constructed a chimeric gene that included the 5% flanking region from the P gene for the small subunit of ribulose biphosphate boxylase containing transcription regulatory sequences, the coding region of a bacterial chloramphenicol acetyltransferase gene as an early selectable gene, and a 3% flanking region from the nuclein synthesis gene containing signals, both for termination of transcription and for polyadenylation of mRNA. Normally, the gene for the small subunit ribulose biphosphate carboxylase is expressed only in green or photosynthetic tissue as expected. The chloramphenicol acetyltransferase gene under the regulatory control of this DNA sequence was also expressed only in photosynthetic tissues. This work provided one of the first demonstrations that, despite the complexity, plant promoters could direct the transcription of heterologous proteins accurately with the tissue specificity. Since this study was done, researchers have used a wide variety of plant protein promoters to direct tissue and developmental specific heterologous gene expression in transgenic plants. Moreover, this higher level of insecticidal toxin synthesis is directly correlated with the increase in insecticidal activity. In another approach to increasing the expression of the protoxin, one group of researchers expressly the full modified protoxin gene under the control of the promoter, the gene that codes for the small subunit of the plant enzyme ribulose biphosphate carboxylase, and downstream from the chloroplast transencaptate sequence of this enzyme. So that the overproduced protoxin become localized within the chloroplast. This strategy led to a very high level of expression, nearly 1% of the total leaf protein of the insecticidal protoxin. Other researchers have introduced an insecticidal protoxin gene directly to the chloroplast DNA of the host plant. Right, we're on to a milestone note: light-inducible and chloroplast-associated expression of a chimeric gene introduced into Nicotiana tabacum. courtesy to L. Herrera Estrella, Van de Broek, Menot, Herrera, Estrella, Van Montagu, Shell, Timko, and Cashmore. It was as post published in Nature 310, pages 115 to 120, year 1984. And after researchers had established the entire plasma system as an effective means of transforming many different plants, their attention turned to the development of procedures for the expression of foreign genes in plants. Initially, most of the genes that were introduced into plant cells were under the transcription control of either the relatively strong constituted 30S promoter from cauliflower mosaic virus or the nearly as strong constituted promoter for the nopalin synthase gene, that is encoded within some tDNAs. However, the development of plants with useful new and modified traits often requires that a specific protein be expressed only in certain tissues, example, leaves or roots, or only at certain times in the life of the plant, example during early seedling development, fruit formation, or high temperature stress. As a first step toward the development of plants that expressed foreign genes in a tissue-specific manner, Herrera Estrella et al. Constructed a chimeric gene that included the 5' flanking region from the P gene for the small subunit of ribulose biphosphate carboxylase containing transcriptional regulatory transferase, acetyltransferase, and the gene as an early selectable gene, and the 3' flanking region from the nopalin synthase gene containing signals both for termination of transcription and for polyadenylation of the mRNA. Normally, the gene for the small subunit of ribulose biphosphate carboxylase is expressed only in green or photosynthetic tissue or photosynthetic tissue as expected. The chloramphenicol acetyltransferase gene under the regulatory control of this DNA sequence was also expressed only in photosynthetic tissues. This work provided one of the first demonstrations that, despite their complexity, plant promoters could direct the transcription of heterologous proteins accurately with the tissue specificity. Since the study was done, researchers have used a wide range of plant promoters to direct tissue and development-specific heterologous gene expression in transgenic plants. Meanwhile, moreover, this higher level of insecticide toxin synthesis was directly correlated with increased insecticidal activity. Another approach to increasing the expression of the protoxin, one group of researchers expressed the fully modified protoxin gene under the control of the promoter for the gene that codes for the small subunit of the plant enzyme ribulose biphosphate carboxylase and downstream from the chloroplast transit peptide sequence of this enzyme, so that the overproduced protoxin became localized within the chloroplast. This strategy led to a very high level of expression, nearly 1% of the total leaf protein. Of the insecticidal protoxin, other researchers have introduced an insecticidal protoxin gene directly into the chloroplast DNA of the host plant. The The Bacillus thuringiensis protoxin gene was integrated into a specific site of the chloroplast DNA by constructing a vector that contained the protoxin gene flanked by two single-copy chloroplast genes. Integration of the introduced genes occurs by homologous recombination. Once integrated into the chloroplast DNA, a protoxin gene under the transcription control of a strong chloroplast promoter may be expressed at high levels, so the protoxin may compose as much as 2 to 3% of the total soluble protein in the leaf, yielding a very high level of insecticidal activity. In addition, even this level of foreign protein expression could be dramatically increased by 10 to 20-fold by co-expressing as part of the same operon that was introduced into the chloroplast DNA, a beta thuringiensis gene that encodes a chaperone protein that facilitates the correct folding of the insecticidal protein protoxin. Integration of the beta thuringiensis protoxin gene into chloroplast DNA has a number of potential advantages of inserting it into the chromosomal DNA. First, the protoxin does not have to be modified because the chloroplast transcription and translation apparatuses are typically prokaryotic. Second, because there are many chloroplasts per cell and many copies of chloroplast DNA per chloroplast, the protoxin gene is present in multiple copies and therefore is more likely to be expressed at a high level. Third, in most plants, chloroplasts are transmitted only through the egg and not through pollen, which means that plants receive all of their chloroplast DNA from their female parent. Consequently, there is no risk of unwanted transfer of the protoxin gene to other plants in the environments by pollen. The disadvantage of expressing the beta thuringiensis protoxin in chloroplasts is that insects that attack stems or fruit will not encounter the protoxin, since the tissues does not have any chloroplast. To date, some form of the gene for the protoxin has been introduced and expressed in a wide variety of plant species, including alfalfa, apple, broccoli, cabbage, canola, corn, maize, cotton, cranberry, eggplant, grape, hawthorn, juneberry, peanut, pear, poplar, potato, rice, rich bagger, soybean, spruce, sugarcane, tobacco, turner, walnut, white clover, and white spruce. Following several seasons of successful field trials, these transgenic plants were approved for a commercial release in the United States, Canada, and Argentina, and large-scale growth of the plants in the field began in 1996. Although insect populations still have to be monitored to keep track of the frequency of resistant organisms, the use of crops expressing B. thuringiensis insecticidal proteins has already exceeded the length of time that it typically takes for resistance to arise in insects to conventional pesticides. A number of transgenic plants that express an insecticidal toxin or protoxin are currently being used commercially. It is estimated that worldwide in 2007, farmers planted approximately 40 million hectares of transgenic Bacillus thuringiensis insecticidal protein containing crops. This technology has more than lived up to the hopes and expectations of scientists, notwithstanding the initial concerns about the technology. Especially in Europe, this approach to crop protection has gained widespread acceptance throughout much of the world. Other strategies for protecting plants against insects. No single beta thuringiensis protoxin is effective against a broad spectrum or broad range of insect species. This may limit the overall usefulness of these protoxins. However, plants have evolved general insect defense mechanisms that are sufficient for plant survival, but not always effective enough to know the damage to a level that will be acceptable for crop plants. For example, some plants produce protease inhibitors that, when ingested, prevent the feeding insect from hydrolyzing plant proteins, thereby effectively starving the predator insect. Consequently, it seemed reasonable to isolate a plant gene for a protease inhibitor, add a strong promoter, and create transgenic crop plants and produce sufficiently high levels of the protease inhibitor to reduce damage from insect predation. Protease inhibitors. In one study, researchers isolated a clone that encodes cowpea trypsin inhibitor from a complementary DNA, cDNA clone bank by using a chemical synthesized DNA probe based on the amino acid sequence of the cowpea trypsin inhibitor protein. The full length of cDNA was subcloned onto a triplasmid binary cloning vector and introduced into a strain of A tumefaciens carrying a disarmed triplasmid. Acetyltransferase gene, MS3' end. So that contained active WRKY genes. Following A tumefaciens infection, tobacco leaf discs with its vector cells that incorporated the cloned DNA were selected for growth on kanamycin, and transgenic plants were regenerated. The damage caused by Helianthus beressens tobacco budworm larvae to transgenic plants that express more than 2 milligrams of cowpea trypsin inhibitor per milligram of protein was significantly less than the damage inflicted on non-transformed plants. Cowpea seeds that contain approximately 2 milligrams of inhibitor per mg of plant protein are non-toxic to either animals or humans. However, if the amount of protease inhibitor produced by a transgenic plant is determined to be a potential hazard, then it is possible to limit the expression of the protease inhibitor to the plant tissues. That the major insect pests prefer but that are not used as food by humans or animals. In other words, a cloned protease inhibitor gene could be active in the leaves and roots of a plant, but not in the commercially valuable fruit. Introduction of the protease inhibitor to gene provides rice plants with protection against the pink stem borer, Sesame inferens, a major insect pest of rice. Infestation of rice plants by pink stem borers cause severe damage to the plants, often resulting in the hollow stem and dead particles with no seeds. A plasmid carrying the potato proteinase inhibitor 2 gene under the control of its own promoter and transcription termination region was constructed. The plasmid also contained the first intron from the rice actin gene inserted between the promoter and the potato protease transferase 2. And coding region. This construct was introduced into rice suspension cells by microprojectile bombardment, and transgenic plants were generated. When pink stem borer larvae were artificially supplied, 70 to 100% of the wild-type plants were severely damaged by insect predation, while only 15 to 20% of transgenic plants were damaged, since plant protease inhibitors are common components of both human and animal food and are readily inactivated by cooking. The introduction into new crops can be regarded as safe. Another strategy that is designed to increase the effectiveness of relatively low levels of beta-thuringiensis insecticidal toxin activity entails combining the toxin with a serine protease inhibitor. In laboratory trials, investigators found that when the amount of purified beta-thuringiensis insecticidal toxin that causes minimal insect mortality was mixed with a low concentration of protease inhibitor, the insecticidal activity of the mixture was 20-fold greater than that of the beta-thuringiensis protoxin alone. To test whether this scheme would function in transgenic plants, a DNA fragment that encoded the fusion protein of both the protease inhibitor and truncated toxin was constructed. Transgenic tobacco plants that produce small amounts of this fusion protein were protected from insect attack. Alpha amylase inhibitor, another way of imparting insect resistance to susceptible plants entails using a gene that encodes an alpha amylase inhibitor. The cowpea weevil, Calosebruchus maculatus, and a zucchini bean weevil, Calosebruchus chinensis, are seed-feeding beetles that cause considerable economic loss of these legume crops, especially in developing countries. When larvae of these insects are fed a diet that includes the common bear insect, that is Physiolus vulgaris, insect growth is inhibited. This growth inhibition is attributable to the presence of alpha amylase inhibitor in the seed protein of the common bean, Accordingly, the gene for the alpha amylase inhibitor from the common bean was isolated, placed under the transcription control of the strong seed-specific promoter for the bean phytohemagglutinin gene, and used to transform pea plants Pisum sativum. Peas are usually quite susceptible to damage by both cowpea weevils and azuki bean or weevils. However, transgenic pea plants that express the alpha amylase inhibitor were resistant to both of these insects. The level of resistance to cowpea weevils was found to be proportional to the amount of alpha amylase inhibitor for the transgenic plants produced. Now on to the cholesterol oxidase. Another approach to developing insect-resistant transgenic plants makes use of bacterial cholesterol oxidase gene. Cholesterol oxidase, which is present in a range of different bacterial genes, catalyzes the oxidation of 3-hydroxy steroids to ketosteroids and hydrogen peroxide. This enzyme is commonly used in assays to determine the levels of cholesterol in human serum. Low levels of the enzyme have a high level of insecticidal activity against larvae of the boll weevil, Anthenomys grandis. A common and economically important insect is Coleoptera, pest of cotton, and have low levels of activity against some Lepidoptera pests. Cholesterol oxidase probably acts by disrupting the insect's midgut epithelial membrane, thus killing the insect. Cholesterol oxidase gene encoding a protein with a molecular mass of approximately 55,000 Daltons with a length of 504 amino acids plus a leader peptide of approximately 5,000 Daltons, 43 amino acids, was isolated from a strain of streptomyces and cloned into a vector under the control of a plant virus, figwood mosaic virus, promoter, and a termination sequence from the 3' region of the A-tumor patient's nopalin synthase gene. When this construct was introduced into tobacco cell protoblasts, the transformed cells actively express the cholesterol oxidase when the gene is introduced into cotton plants on a compound scale, on a commercial scale, either by itself or in combination with genes or other biological insecticides. It should be an effective means of protecting plants against damage from insect predation. Vegetative insecticidal toxins. In addition to the well-characterized WRKY insecticidal toxins, about 350 of which have been identified, beta-thuringiensis produces secreted insecticidal protein during its vegetative growth stage. To date, 2 major groups of vegetative insecticidal proteins, VIP, have been identified. One group consists of the proteins VIP1 and VIP2, which are not toxic, to Lepidoptera, and VIP3, which target several other Lepidopteran pests. The less well characterized VIP proteins may act synergistically with cryoproteins to kill their target insects, providing a double-barreled approach to insect toxicity so that it is extremely difficult for susceptible insects to develop resistance. It would therefore be advantageous if transgenic plants expressing both cry and VIP proteins could be created. As a first step, researchers shuffled the two major domains of two VIP3 proteins and displayed the high activity. One of the hybrid VIP3 proteins displayed the high activity of the four proteins against family armyworms, fall armyworms, cotton bollworms, silkworms, moreover. The VIP3 ACHA construct was toxic to the strain of cabbage looper that was resistant to the well-characterized beta-3-engineered insecticidal protein. The chimeric toxin enriches the diversity of these construct that can be used together with conventional cryoproteins to generate transgenic plants that are highly unlikely to select for resistant insects. Other proteins. The activities of several other proteins have been utilized in an effort to protect plants from insect predation. For example, some of the lectins, that is carbohydrate binding proteins. Found the seeds and storage tissues of a variety of plant species are toxic to certain species of insects, while many plant lectins are toxic to mammals as well as insects. The lectin from the snowdrop plant Galanthus nivalis is toxic only to insects. With this in mind, the snowdrop lectin gene has been introduced into approximately a dozen different plants, with the result the plants that express this protein were damaged by aphids to a lesser extent than non-transformed plants. However, although the snowdrop lectin significantly lowered the amount of leaf material eaten, insect mortality was only slightly increased. And the gene encoding the enzyme tryptophan carboxylase from periwinkle Catharanthus roseus is expressed in tobacco. The plants are protected from damage by the whitefly Bemisia tabaci. While the precise mechanism of this protection is unknown, it has been suggested that the tryptamine that is produced by this enzyme following the decarboxylation of tryptophan is used in the production of insect-inhibiting phytoalkaloids. Should I say that? Plant alkaloids. The gram-negative bacterium Photorhabdus luminescens produces a 283 kilodalton protein, toxin A, that is highly toxic to a variety of insects. When the protein was expressed in transgenic Rhabdosa thaliana, proteins in amounts greater than or equal to 700 nanograms per milligram of extractable plant protein, it was found to be highly toxic to the tobacco hornworm, as well as the southern cotton rootworm, with insect mortality typically 100%. Finally, transgenic corn plants that express avidin, a glycoprotein isolated from chicken eggs that binds the coenzyme biotin with the extremely high affinity, caused biotin deficiency that led to stunted growth and death in a number of different insect species. Importantly, the levels of avidin that are toxic to insects are not toxic to mice, suggesting that protecting plants with an abundant transgene is not necessarily a problem for humans. RNA interference ingestion is a microinjection of double-stranded RNA into some worms and insects has been used to silence genes in these organisms. These gene silencing works through the generation of RNA interference RNAi. In one study, when 290 different double-stranded RNAs thought to encode essential or important functions were fed at a low level to larvae of the western corn rootworm, WRKY, LeConte. Growth was significantly inhibited with 67 of the RNAs. 40 of the double-stranded RNAs had 50% lethal concentrations of greater than or equal to 5.2 nanograms per square centimeter. Based on these results, a transgenic corn expressing one of the double-stranded RNAs targeting the transcript for vacuolar ADVase was constructed. The transgenic plants were protected against the western corn rootworm to an extent comparable to the protection afforded by a beta-thuringiensis transgene. The demonstration that it is possible to produce RNA in Coleoptera following oral delivery of double-stranded RNA is an important first step in the development of a completely novel approach to developing a wide range of insect-resistant plants, including resistance to some insects that are refractory to the beta-thuringiensis insecticidal toxin, and a variation on the above-mentioned strategy. Another group of researchers first identified the mechanism that the cotton bollworm uses to protect itself against the compound gossypol, which is produced by cotton plants to prevent insect predation. Gossypol is a yellow polyphenolic aldehyde that permeates cells and acts as an inhibitor of several of the insect's dehydrogenase enzymes. It's been used as a male oral contraceptive in China. It possesses antimalarial properties and may have anti-cancer properties. The cotton bollworm protects itself from the toxic effects of gossypol by inactivating the gossypol with the enzyme cytochrome P450 monooxygenase. That transgenic plants are constructed to synthesize an RNAi molecule that would silence the insect's gene for the cytochrome P450 monooxygenase by preventing the expression of cytochrome P450 monooxygenase. The insect was exposed to the full toxic effects of the plant-produced gossypol so that it was either killed or at least debilitated, and the allelochemicals and the damage to the plant was limited. Plants produce a myriad of allelochemicals to protect themselves from insects, and many insects have developed strategies to overcome the toxic effects of these compounds. Therefore, the mechanism used by insects to overcome the toxicity of the plant-produced compounds are attractive targets for developing insect-resistant plants in the future. We're on to another milestone note. Arabidopsis thaliana. This Thalcris is a small weed in the same family of Brassicaceae as canola, mustard, and broccoli. It is native to Europe, Asia, and northwestern Africa. Thaliana is popular with insects as a model organism in plant biology and genetic studies. It has one of the smallest genomes at 7 × 10⁻⁷ base pairs. It's similar to the size of the yeast genome, which is approximately 1.5 × 10⁻⁷ BP of any flowering plant, which makes it readily amenable to molecular genetic studies. The small size of its genome has made a thaliana useful for the generation and selection of mutants, and it was the first plant genome to be sequenced in 2000. The plant's small size and short life cycle are also advantages for research. Laboratory strains of A. thaliana take about 6 weeks from germination to mature seed. The small size of the plant is conventional for cultivation in limited space, and it produced many seeds. An individual plant can produce several thousand seeds. Plant transformation Arabidopsis is straightforward and has become a routine procedure in many laboratories, using 8 mutations to transfer DNA to the plant genome. The current Arabidopsis transformation protocol termed floral dip involves dipping a flower into solution containing agrobacterium, the DNA of interest, and a detergent. This method avoids the need for tissue culture or plant regeneration. The idea is that some of the agrobacterium cells will transfer the tDNA containing the target DNA into reproductive tissue of the plant. As a consequence of the above-mentioned traits and the relative complexity of most of the plants, Arabidopsis has become the E. coli of the plant world. There is little doubt that insects have the gene with genetic potential to develop resistance to beta-thuringiensis insecticidal toxins. And the more that beta-thuringiensis insecticidal toxins are used, the greater the likelihood that populations of targeted insects will accumulate resistant individuals. Experimental strategies have been devised to prevent transgenic plants that express the Bacillus thuringiensis rhodopsin gene from acting as a selection against agents for resistance to resistant insects. In one approach, the expression of the insecticidal toxin in transgenic plants was limited to a short period. The gene for B. thuringiensis protoxin was cloned downstream of the promoter of a gene from tobacco called the pathogenesis-related protein 1A, PR1A gene. The expression of the PR1A gene is a part of a natural defense mechanism that combats pathogens. The PR1A gene is normally induced by any one of a variety of pathogenic organisms or by chemicals such as salicylic acid and polyacrylic acid. When transgenic plants with the Bacillus thuringiensis protoxin gene under the control of the PR1A promoter were treated with a chemical inducer, they synthesized detectable levels of insecticidal toxin within 1 day of application, which protected the genes against one insect attack. Therefore, it is conceivable that the protoxin could be induced by the administration of an inexpensive and safe chemical inducer only when it is required during the growing season. When insect larvae are feeding, such periodic production should lower the selection pressure of a resistant bacteria insects. One approach that might increase the insecticidal effectiveness of beta-thuringiensis expressed in transgenic plants and also decrease the development of insect resistance is to fuse the bacterial insecticidal gene with another protein that increases the binding of the insecticide to the target intestinal cellular receptor. With this in mind, the fusion protein consisting of an N-terminal B-thuringiensis insecticidal toxin and a C-terminal peptide consisting of the non-toxic beta chain of the protein-rich ricin was constructed. Ricin is a protein toxin that is extracted from castor beans. Consists of an A chain of 267 amino acids that contains the toxin activity, and a B chain of 262 amino acids that contains the toxin activity, and a B chain that is catalytically inactive but serves to mediate entry of the complex into the cytosol. The B-thuringiensis insecticidal toxin binds to a receptor located within the membrane of the insecticide gut. Normally, since each insecticidal toxin interacts with a single receptor, the loss of modification of the receptor leads to resistance to the insecticidal toxin. And however, since the ricin B chain binds with the very high affinity to N-gacetyl galactosamine residues, which are adjacent to the B-thuringiensis insecticidal toxin receptor, the fusion protein has two separate and independent means by which it is targeted to the receptor. With this fusion protein, it becomes extremely unlikely that both targeting mechanisms will cease to be effective at the same time. It has been suggested that this approach may be most effective in field situations where it is difficult or possible to implement a spatial refugium strategy such as transgenic rice. A number of additional strategies designed to prevent the development of insects are resistant to the beta-thuringiensis insecticidal toxin has been developed, and they include the following: using spatial refuge strategies where a certain fraction of each farmer's land, generally around 20%, is planted with a non-transgenic crop, where the remainder of the land is planted with a transgenic version of the same crop, expressing a high level of the B-thuringiensis insecticidal toxin. The idea behind this strategy is that the very small number of insects that are able to survive on the transgenic insecticidal crop, a high dose of toxin called 99.9% of susceptible insecticides, will mate with the much larger number of toxin-sensitive insects from the transgenic crop. Thus, the gene for resistance is effectively diluted. A high dose of the toxin kills 99% of heterozygotes, and the pest pollution remains sensitive to the insecticide. Using two or more different B-thuringiensis insecticidal toxins, sometimes called gene stacking, or fusing portions of the active regions of two different toxin genes to generate novel hybrid protein insecticidal toxins. Also see chapter 116. This approach assumes that the resistance of two control methods is much less likely to develop simultaneously. This approach has been found to be effective in the field where it combines spatial refugia. And the next point will be transforming plants with a B-thuringiensis insecticidal toxin gene and in the form of biological insecticide, example an alpha-amylase inhibitor gene. This also assumes that resistance to two control methods is much less likely to develop simultaneously. Spraying low levels of chemical insecticides at the same time, the transgenic in plants expressing B-thuringiensis insecticidal toxin gene are used. This also assumes that resistance to two control methods is much less likely to develop simultaneously. And the last point would be spraying low levels of chemical insecticides. At the same time, the transgenic plants expressing B-thuringiensis insecticidal toxin gene are used. This also assumes that resistance to control methods is much less likely to develop simultaneously. The insect resistance management strategies that have been used up to now appear to have been successful. For example, in one large study, researchers monitored the level of resistance of pink bollworm, Betaine gossifera, to B-thuringiensis in cornfields over the course of 8 years, from 97-2004. They found that so this lecture should be over in a minute from now. I'll just catch up the points in the last para. And it was found that the resistance did not change over this period, attributed to this result of the use of refugia, the recessive inheritance of the resistance, the fact the resistance that developed was incomplete, and the fitness costs associated with the development of insect resistance. And now, the amount of land that's devoted to the growth of transgenic crops that express A, B genes insecticidal toxin continues to increase rapidly worldwide. Thus, a responsible approach dictates that we can continue to be careful and vigilant in avoiding the development of insect resistance so as not to waste this resource. So make notes on these lectures that I'm giving you. Research as much as you can. Get prepared. Please do not commercialize anything. Set your doubts. Send all your doubts and for clarifications to my comment section. I'll be back with you soon. Bye.